a B minimum. And so I know it's always a hard decision, but and to make it even harder, I'm not going to get those. Obviously, I don't have the lab practice. So yeah. So just remember, you guys. I think it's Sunday. I believe that's the last day to drop. If you're thinking about it, double check on the Saxony website just to make sure. And then another thing that came up, and I wanted to apologize for causing any additional stress in your lives. There was like a virus homework quiz posted on D2L. It was just for practice. I stole it from the class night, and it was just for practice, kind of like just a worksheet. So it's not due, yeah. Okay, and then what we're going to do today is we're going to launch into part 2 of viruses. We're going to look at some specific viral pathogens, viruses that invade humans and other organisms and cause significant disease. And you guys might recall that hopefully I mentioned, I'm actually going to revert to the order that we look at these sample viruses. The way the viral pathogen PowerPoint is set up, it starts with DNA viruses. But what I'm going to do this semester, we're going to go ahead and start with RNA viruses because they tend to be a little bit more complicated. And the RNA viruses that we're going to touch on today, probably most molecularly on influenza virus, and then we'll touch very lightly on rabies virus, West Nile virus, and then we'll have, next week, we'll have quite a bit on HIV. The 2 handouts from today, one of them is on influenza virus and host and tissue tropism, which we'll be discussing. And then the other one is a diagram that I stole from Scientific American. It has to do with HIV replication. And on the back, there's some information on target cells of HIV, and then a table on antiretroviral drugs, which we will be discussing next week. But it does help if maybe you take a look at the HIV replication at a time. Okay, so what we're going to do then is we're going to launch into our first RNA virus. And again, we want to remind ourselves that RNA viruses, since they use RNA polymerase to copy their genetic information, RNA polymerase, as we know, don't proofread, don't edit. RNA polymerase makes lots of mistakes copying viral RNA, and consequently that means that RNA viruses have a high potential for what? Mutation, right? And we always need to keep that in the back of our mind that they have this high potential for mutation. And we see that just in looking at the number of different RNA viral families. There's around 50 viral families, RNA viral families. And you'll recall that when we looked at the number of DNA viral families, there's only about 7. And again, we would propose that the high mutation rate is one explanation for the greater diversity amongst RNA viruses. So we're going to take a look at, as I mentioned, a few RNA viruses. The orthomyxoviridae, this is a virus family. The one we'll focus on is influenza virus. We'll look at rabies virus. We'll look at one of the arcoviruses. Arcoviruses are arthropod-borne viruses. We're going to focus on West Nile virus. And then we will talk about HIV. And then we'll finish with a brief discussion of naked RNA virus of poliovirus. Okay, so influenza virus. Last semester, I went down to the state public health department. They have a big reference lab down in Vallejo, and we was just talking about what are the biggest concerns, public health concerns, and what kept coming up over and over and over again was influenza virus. So certainly there's many other microbial pathogens of huge public health importance, HIV, for example, of huge public health importance. But influenza virus appears to be the one that the public health officials are most worried about causing widespread epidemics and potentially pandemics, worldwide epidemics. So I thought it would be worth our while to spend a little extra time on influenza, understand why folks are so worried about the pandemic possibility. Okay, so influenza virus, it is an RNA virus, and it has a cool genome in that the genetic information is in so it's almost like the RNA is an 8 little RNA chromosomes. You could almost think of them. And this segmented genome makes it possible for influenza viruses to mix genetic information. So we're going to see that a lot of influenza viruses are actually recombinants. They're carrying genetic information from more than one influenza strain. The influenza virus, the good news with influenza virus, it is an envelope virus. Okay, so these are two different artist cartoons of possible influenza viruses. So this outermost layer then is the viral envelope that's been stolen from our cell membranes. And you'll recall that these envelope viruses, before they steal our cell membrane, they have to modify our cell membranes by inserting viral proteins in the membrane. And there's two very important influenza viral lipoproteins. Lipoproteins meaning a little carbohydrate or sugar is going to add to the protein. And the first one is called hemagglutinin. It's often referred to as the H antigen. And it was called hemagglutinin, it literally means blood clumper, because it was discovered when you mix influenza virus with red blood cells, the influenza viruses can attach to the surface of the red blood cells, cross-link them, and form these clumps of red blood cells. Now, that's not the natural function of hemagglutinin. It's not the function it was designed for. The hemagglutinin is the influenza adhesin. What is an adhesin? Right, it's the molecule on the surface of the virus that permits it to attach to a host cell. And we want to remember, you guys, with viruses, they have to attach to the surface of the host cell before they can infect it. Okay, so the hemagglutinin then, it is the viral adhesin, and it binds to host cell surface sialic acid. And we'll come back and talk a lot more about the different types of sialic acid. Okay, so we would say that the host cell surface receptor then is going to be the sialic acid. Now, the hemagglutinin is, as we mentioned, it's made out of proteins. And proteins are excellent antigens. Antigens are substances which trigger an immune response. And an example of an immune response would be antibody production. All right, so the hemagglutinin is often called the H antigen because it's going to trigger lots and lots of antibodies to be produced. And indeed, antibodies made against the hemagglutinin are considered protective. Why do you think, if we have high levels of circulating antibodies against the H antigen of an invading influenza virus, why do you think those antibodies would be protective? Which step in influenza infection of our cells do you think those antibodies would block? You guys need a second cup of coffee. I understand totally. Okay, so we have antibodies coating the hemagglutinins. What can hemagglutinins do? Attach to our cells, right? So we call antibodies that block attachment. Do you guys remember what we call those types of antibodies? Neutralizing antibodies. Okay, so antibodies that we make against the H antigen are considered neutralizing antibodies because when they attach to the H antigens, they'll block the ability of the influenza virus to attach to our cells. And remember, if the influenza virus can't attach to our cells, they can't infect our cells, we're not going to get sick. Now, what we're going to discover, one of the huge problems with influenza viruses, is that H antigens are constantly mutating, they're constantly changing, and that's going to be a theme we'll come back to. All right. The second very important influenza envelope lipoprotein is called the N antigen for neuraminidase. And the neuraminidase is a really interesting protein. Okay. As a protein, it too triggers really strong antibody production, so it's often referred to as the N antigen. And there's one confirmed function of neuraminidase neuraminidase, and a second function that may help influenza in the infection process. So the first function is that the neuraminidase will hydrolyze sialic acid to help newly replicated influenza viruses escape from the host cell and to prevent clumping of influenza virus. And what we'll do, you guys, if we can just hold on to this, there's an electron micrograph and some diagrams that will help us understand exactly what this neuraminidase is doing. Okay. The second function that some folks believe is also important is they think the neuraminidase may also help hydrolyze mucus overlying the epithelial cells of the host organism. So, for example, with human influenza strains, we are infected through inhalation, and the cells that the influenza virus then are going to infect would be the epithelial cells of the respiratory tract. And you guys recall, especially in the upper respiratory tract, we have this beautiful innate defense, the mucociliary escalator, this thick mucus blanket that covers the respiratory epithelial cells that acts as microbial flypaper. Right? So influenza viruses can get stuck in that mucus blanket. And then we have those beautiful ciliated cells in the trachea that eat together to move that mucus blanket up to the back of our throat, where we can spit it out with the entrapped microbes, or we can swallow it where they'll be destroyed in the stomach. Well, that mucus blanket not only acts as microbial flypaper, it actually acts as a barrier between the invading pathogen, the virus, and the surface of the epithelial cells. So some people think the second function of neuraminidase is it hydrolyzes the protein portion within the mucus and permits influenza virus basically to eat its way through the mucus blanket so it can reach the surface of the respiratory epithelial cell so that it can then bind and infect the cells. Okay. So the neuraminidase or N antigen also will produce antibodies, maybe not as protective as the hemagglutinin. The most important antibodies that we can make are against the hemagglutinin, the adhesive. Okay, so in this particular cartoon, we see the envelope. These little red spikes represent the hemagglutinins, and the little blue spikes here are the neuraminidase. Okay, and in this second picture here, we got the hemagglutinin and the neuraminidase. Now, not to worry, you guys, there's another protein called the M protein that lines the inside of the envelope. Not to worry about that. But inside, then we have the RNA. And I'm going to switch to this picture, you guys, because it's a little bit more clear that the RNA of the influenza is in these 8 segments. So the RNA segments are covered with proteins. So this makes up the nucleocapsid. The nucleocapsid has a helical structure. Okay, so we have 8 segments, and each segment carries a gene for a different protein. Okay, so it really is a little bit like little chromosomes. Okay. All right, so we want to remember that because the influenza virus is enveloped, once it's shed from the host, any damage to the envelope makes them non-infectious. So if the influenza virus were too dry, to be exposed to sunlight, to chemicals, to soaps, it will damage the envelope and they're no longer infectious. So that's good news. Returning to the H and N antigens, it's been discovered that there's many different types of H and N antigens. So when a new influenza virus is identified, the first thing folks want to know is what is its H subtype and its N subtype. So as an example, we can see here that some influenza viruses are called H1N1. We have other ones that are called H2N3. Okay, and just with this, I want to give you just the tiniest bit of classification with these influenza viruses. So it turns out that there are 3 groups of influenza viruses. There's group A, B, and C. So group C, these usually cause very mild disease. They tend to be very stable in that they don't change very often. And so we don't really worry about group C influenza viruses. They're just not that big a deal. They're not even included in the annual influenza vaccine. Okay. Now group Bs are a little bit, they can cause serious disease. We can see some antigenic variations, so I'll just put some variability. But the group Cs usually, they primarily have humans as their primary reservoir. So in the annual influenza vaccines, there's usually one group B influenza included in the annual vaccine. The group we're really worried about are the group A influenzas. So our lecture is going to totally focus on the group A influenzas. These guys are highly variable, high variability in antigens. These guys can cause they have potential for serious disease. And another very important feature of these guys, many of the group A's have non-human animals as reservoirs. So humans and animal reservoirs. And we're going to see that the strains carried by these animal reservoirs will provide a lot of the genetic variability that we see being produced into some of the new evolving human strains. Okay, so this is going to be our primary focus, are the group A strains. And in the annual influenza vaccine, always 2 group A strains are included. So we call these groups. And then when we use the H and N antigens, these are referred to as the subtypes. And just to give you an idea, the last pandemic we had was the 2009 pandemic. The pandemic meaning worldwide epidemic. Okay. It was a group A. It was a group A influenza virus, and the subtype was H1N1. And the reason, one of the reasons public health officials got so worried when this pandemic H1N1 evolved is that this is the same description to the horrific 1918-1919 pandemic influenza that killed, they don't even know how many people it killed because so many people died, records weren't kept. But the low end of the estimates are 25 million people died, up to 100 million or possibly more. Okay, so at the time of that epidemic, it appeared that pigs became infected. So it appeared that swine or pigs got infected. So there was a swine influenza outbreak that seemed to coincide with this horrible pandemic. And another reason that folks were worried is that there was some suggestion that this pandemic strain may have originated in swine. So it was mirroring that horrific 1918 pandemic, and that's why people were so worried. They came up with a vaccine against this strain, and the vaccine has been included in the most previous annual influenza vaccine. So if you got vaccinated against influenza this year, you were vaccinated against this pandemic strain. So just a little bit on the classification. All right, now, as we said, a huge issue with influenza viruses is how quickly they can change, how quickly their H antigens can change. So for example, if we get infected with one strain of influenza virus this year, we may be neutralizing antibodies against that strain. Next year, we might get infected with an entirely different H antigen influenza virus, and we won't have any protection against it. So this is a huge issue. One of the reasons why we have to get new vaccines every year. One of the reasons we don't acquire lifelong immunity against against influenza viruses. Now, there's two ways that influenza viruses can change antigenically, and the two mechanisms are antigenic drift and antigenic shift. Antigenic drift we're actually familiar with already. Antigenic drift are tiny changes in the H and N antigens that are a product of mistakes that RNA polymerase makes when it's copying the influenza genetic information. So antigenic drift are these small changes in the H and N antigens that are a result of mutations. So when the RNA polymerase copies the genes for the H and N proteins, it makes mistakes. There's little changes in the amino acid sequence, so it makes little changes in the H and N antigens. And you guys are going to laugh at me, but I always have trouble, remembering the difference between drift and shift. So my little way to remember drift is I think of driftwood on, like the ocean or a lake, and it's just slowly drifting along. Little changes in positions, little changes in the H and N antigens, right? Okay. Now, in contrast, this antigenic shift, this is kind of a new concept for us. Antigenic shift are huge changes in the H and N antigens. And antigenic shift is a result of two different influenza strains infecting the same host and infecting the same cell. It's just wild. And as a result of those two different strains of influenza virus co-infecting the same cell, when the new viruses are made, you might get brand new combinations of genes, like a mixture of strain 1 and a mixture of strain 2. So the newly replicated viruses can have brand new combinations of H and N antigens. And that causes huge changes in the H and N antigens. So that's called antigenic shift, big changes. And again, you guys, I always have to come up with some kind of way to remember this stuff. So antigenic shift, I'm like, okay, well, in the old days when we used to have typewriters, there was a shifting mechanism, and it was a big change in position. So I think of antigenic shift like the typewriter, old-fashioned typewriter. So antigenic shift is big change, big change, big change in position, big change in H and N antigens. And it's believed that antigenic shift is responsible for the worldwide epidemics, the pandemics we experience every 20 to 30 years. So we're going to come back and we're going to explore antigenic shift. Just as a quick just as a quick example, you guys, we could come back here and use these two different subtypes of influenza virus. And let's say that a host was co-infected with both these strains, and one of the host cells were co-infected with these two strains. So following replication and assembly, the little RNA segments from the different strains are just packaged at random. So we could end up with newly replicated viruses that are reassortments or recombinants of genes from these two strains. So for example, some of the recombinant virus that are released could be H1N3s. Some of the recombinants could be H2N1s. Right? And the problem with that is often it creates new combinations that humans don't have any immunity against. Okay, so let's take a look here. All right, so this is an electron micrograph. Influenza viruses budding, escaping from an infected cell. And just to get you oriented here, this is the cytoplasmic membrane. This would be the interior of the infected host cell. And this has been colored after the fact. Here are the budding influenza viruses here. You can see them budding. Now, this is really helpful because it helps us to understand one of the functions of the neuraminidase. Okay? So you guys remember that the influenza virus is binding to sialic acid receptors on the surface of the host cell. Right? And in the budding process, they're stealing that cytoplasmic membrane to make their envelope, right? So what is on the envelope? Sialic acid, right? And as a consequence, these newly replicated influenza viruses, since they have their H antigens present, right, the hemagglutinin, the hemagglutinin of the viruses will bind to the sialic acid on their neighbors' envelopes, right? And what would that cause? Clumping, right? Clumping. And when viruses clump, it's much harder for them to move to other cells and infect those neighboring cells, right? And so the one function for the neuraminidase, it's also on the surface, is to hydrolyze, destroy the sialic acid on the envelope of the influenza viruses so they won't clump together. That will increase their escape from the host cell. And there's also the possibility, like this little guy right here that's budding away, what could happen is this guy's hemagglutinins, in theory, could bind back onto the host cell here. So it's possible that neuraminidase are also hydrolyzing some of the sialic acid in the area where the viruses are budding from. Because it would make, I mean, to me, if I was a virus, I wouldn't want to reinfect this cell because maybe this cell is going to die pretty soon. You know, I might not be able to complete my replication cycle. So it makes to me kind of like survival evolutionary sense that you wouldn't want to reinfect the cell that you just escaped from because that cell might be on its way to dying. Okay, so that neuraminidase function, very cool stuff. You guys perhaps have heard of Tamiflu. So Tamiflu, its target is the neuraminidase. It knocks out the function of the neuraminidase, and thus that will decrease the ability of the influenza viruses to escape from our cells. It will decrease their ability to spread to other cells and should really limit the damage and limit viral replication. Yeah, so again, you guys, I know sometimes we talk about all these little structures and functions, like, oh, why do we have to know this stuff? But it's so cool because by people understanding the functions of all different parts of the influenza viruses, they can come up with new antiviral drugs. Yeah, boy, do we need those antiviral drugs. Okay. Alrighty then. Now, I love this picture, you guys. I had no clue that influenza viruses could infect so many different animals. Okay, so we're going to use this diagram, first of all, to discuss who biologists think were the original hosts of influenza viruses, and then we're going to discuss the spread of influenza viruses from the original host to other organisms. Okay, so out in nature, different influenza strains have been isolated, and they found on those influenza strains, they found up to 15, and it's possibly more now, 15 different H antigens. So out in nature, there's H antigens types 1 through 15, and when they isolate influenza viruses from nature, they find that there's at least 9 different neuraminidase antigens. Now, the host organism that they've been able to isolate influenza viruses with all 15 H antigens and all 9 N antigens have been wild water birds like ducks. And so biologists think probably wild water birds like ducks were probably the original host because we find the greatest variety of influenza viruses in wild water birds like ducks. Now, we think over time what happened was influenza viruses were spread from ducks to different organisms, and then those different organisms spread the influenza viruses to other host organisms. So for example, influenza viruses were spread to non-water birds like quail, were spread to domesticated poultry like chickens, and what we see here are some of the H and N antigen subtypes that have been isolated from, for example, from chickens. These that are in red are the really highly pathogenic avian influenza strains. So birds, or a fancy term for birds is avian. So we talk about avian influenzas. So this H5N1 is the one we're most worried about, and another one of concern that can cause infection in domesticated birds is H7N7. But H5N1 is going to be the one we're going to focus on. It was discovered that the avian influenza H5N1 was transferred from chickens to members of the cat family, felines, and through feeding those cats chicken meat that was contaminated with H5N1. So we have H5N1 infections in cats and tigers in zoos. Zoos have to be really careful what they feed their animals. Also, to me, that was just really fascinating was marine mammals like seals and whales have their own influenza strains. In black, the strains that are in black, these are the strains that are well adapted to that particular host, right? So even marine mammals have them. Horses have their own equine influenza virus strains. Ferrets were a real important lab animal that was used to study some of the like the 1918 strain they use ferrets is one of their lab animals. Of course, humans here. Now, if we take a look at humans, some of the well-adapted human strains that have caused some of the major pandemics in the last century or so, H1N1, for example, as we said, the 1918 strain was an H1N1, and the 2009 pandemic strain was an H1N1. Other pandemics have been caused by H2N2 and H3N2. So these are all considered human influenza strains, well-adapted human strains. But here we see some of the avian influenza strains that have caused infections in humans. And again, we're going to focus on this H5N1. This one is so scary, you guys. Of the humans that have been infected through direct contact with infected birds, the mortality rates have been around 50 to 60%. That means for every 100 people that get infected, 50 to 60 of them die. That is incredibly high mortality rates. I mean, it's just off the charts for mortality rates. The only thing that has saved us so far is that although the H5N1 is highly virulent, there's low transmission human to human. Very few cases of documented human-to-human H5N1 transmission. Okay, and we're going to come back and explore why that could be. But the other organism here, we have a little triangle going here that I want you guys to focus on. The triangle of birds, humans, and here, very important, pigs. Yeah. Pigs, as we can see, if we take a look here, you guys, H1N1, that's a so-called human strain, right? Can infect pigs. Okay, H1N2, H3N2, a human strain that can infect pigs. Okay, and this one, these two are unusual in humans. But the bad news is pigs can be infected with avian influenza and human strains of influenza at the same time. So it turns out pigs are what are called reassortment vessels. Pigs are wonderful hosts for co-infection with human influenza strains, avian influenza strains, and pigs have their own swine influenza strains. Okay, so we want to take a look at, we'll just diagram kind of a goofy cartoon, you guys, of what might happen on the farm. One of the things we're really worried about that could happen. Okay, so I'm going to torture you with my goofy cartoons. Okay, so we're on the farm. And those of you that have ever worked on farms or have family or friends that have farms, you know farmers, even though they're sick, they have to get up and take care of their animals, right? They can't lie in bed and let their animals starve. So we've got a farmer who's infected with a human influenza strain. And these human influenza virus strains tend to have high transmission. They're easily transferred between humans. But most of the human influenza viruses, even though they're easily transmitted, most of them usually don't kill healthy young people like you all, right? You might be sick, might be in bed for a week, might feel lousy for a few weeks, but usually you don't die. The human strains are potentially lethal in our high-risk groups, the very young, the very old, immunocompromised, right? But young people your age, you know, it's like, oh, you know, you might miss an exam or something, but that's going to be, you're not going to die from it. But high transmissibility, right? Okay. And then we're going to have, I better label this. This is a duck, you guys, because nobody will be able to tell it's a duck. Okay, here's a duck. And maybe it's a wild duck. Or maybe it's a domesticated duck. Labels are really important, okay? And what we're going to discover, you know, this influenza story, you guys, is so wild. You know, in humans, influenza virus, it's a respiratory tract infection. Okay, labels are important. This is the duck's intestine, okay? And so I was amazed to find out that one of the primary sites that avian influenza viruses replicate in birds is in their what? In their intestine, yeah. So this is the site of avian influenza. Can also replicate in the bird intestinal tract. Now, if you're working with birds or if you're butchering birds to eat, you have to be careful. presume all the body secretions are contaminated. All the body secretions, in theory, could be carrying avian influenza. But we need to remember that feces is going to be a rich source of avian influenza. And that's something I never think of because I always think of, of it being a respiratory tract infection. So remember, in birds, feces are going to be a rich source. All right, so on this farm, we have some pigs. I love pigs. They're really smart. That looks like a dog. This is a pig, you guys. Pig. Oh, I should put a little, I know, a little curly tail. Yeah, okay. Pig. Wow, that looks like a dog. Okay, all right. All right. So as we saw on this chart, pigs can be co-infected with more than one influenza virus. So co-infected, they're infected by more than one pathogen. So when the human is out there coughing and sneezing and spewing out, I'll use a little white asterisk here for the human influenza virus, coughing in the poor pig's face. The pig inhales the human virus, and I'll just put the human virus there. And the ducks come along and they sit in the feed trough of the pigs, and they're eating the grain and pooping in the grain at the same time. So when the pigs go and eat, they're creating these little fecal aerosols, and they're inhaling the avian influenza as well. So let's put the avian influenza can infect the pig as well. Okay. And as we said, not only are both these human influenza and avian influenza infecting the pig, both those influenza strains can co-infect the same cell. They can co-infect the same cell. And I'm going to fast forward for you guys to there's a diagram here. Okay, so we'll pretend that this is the pig cell, this is the swine cell right here, and here's our two different influenza strains. We'll say this virus 1 is the human influenza strain, virus 2 is the avian influenza strain. Okay, they co-infect the same cell. They undergo biosynthesis, so the human influenza RNA is copied, the avian influenza RNA is copied. And remember, they're on these segments, right? Okay. So when the new viruses are being assembled, it's just haphazard which RNA segments are packaged together. And in some cases, what you'll get, and they say in about 40% of the new viruses released from these co-infected cells, in 40% you'll have recombinant viruses, where there's a mixture of the human influenza genetic information and the avian influenza virus, avian influenza genetic information. So with the artist here has shown this, and this isn't true in nature, guys, but it's just to help us understand. The human influenza is blue, the avian influenza is red, they co-infect, we get reassortment, recombination of genes. So some of the newly released, newly replicated influenza viruses are what? Purple. Just showing a combination of the human influenza and avian influenza genes. So why do we care? Well, let's go back here. And you guys remember we said these avian influenza strains, these guys have incredibly high mortality when humans are infected, right? 50 to 60%. But what about transmission human to human? You remember, and I know we're covering a lot of territory here. Because we said these avian influenza like H5N1, they have really high mortality rates in humans, but the reason we aren't all dead yet is because what? What about human-to-human transmission of the avian strains? Really low. Rarely. So, but low transmission between humans, right? If we get infected, very rarely will we transfer H5N1 between ourselves. Almost all the human cases have been people that have been infected by working with infected birds or bird products. Okay, so, but what about the human influenza strains? High transmission, human to human, but usually low mortality, right? Okay, so what could happen then if we combine genetic information from the human influenza virus, which has high transmission human to human, and we combine genetic information with the avian influenza, which has poor transmission human to human, but has really high virulence? What are we worried about? What are we worried about these recombinants? Why are we so concerned? What could characterize these recombinants? What about transmission between humans? Potential for there to be high transmission between humans, and what makes us so worried about that? What property could they pick up from the avian influenza? High mortality rates, right? So this is what's keeping a lot of public health officials awake at night. This is the nightmare scenario, right? They would get a reassortment, recombination between a highly transmissible human influenza with an avian influenza that has incredibly high mortality rates in humans. This is the nightmare, right, that everyone's so worried about. Okay, now, just to add a little bit more, you guys, it turns out this is probably happening multiple times around the world, and indeed, what we want to add to this is that we want to remember that pigs have their own influenza virus. So when this happens over and over and over again, you can get recombination between human strains, avian strains, and swine influenza strains. So you can end up with these mosaic hybrid influenza viruses. And indeed, that's exactly what they discovered. You can get these mosaic hybrid influenza viruses that carry genetic information from human influenza strains, avian influenza strains, and swine influenza strains. And that's exactly what that pandemic 2009 strain was. When they did the RNA sequencing, yeah, that pandemic 2009 H1N1, when they did the RNA sequencing, it carried genetic information from human, avian, and swine influenza. Okay, so these group A influenza viruses have incredible potential to undergo these recombination events and generate incredible genetic diversity and generate strains that we're worried about that have really high transmission between humans and have really high mortality rates. That's the nightmare. Luckily, this one didn't. Initially, they thought it was causing high death rates in young, healthy people like you, and it turned out when the whole thing was over and they did data analysis, it wasn't true. But that high mortality rate amongst young, healthy people, that was another unusual feature of that 1918 strain. It was young, healthy people that were dying. And so when this 2009 strain hit, and it seemed like it was making a lot of young people sick and causing deaths, everyone was like, oh no, here we go again. Here's the 1918 pandemic all over again. Yeah.
So did they have to kind of just wait for something bad to happen to find out? Like
No, you're absolutely right. I mean, that's why a lot of these folks are losing so much sleep, because they know the potential, but they don't know what that strain is going to look like. And because you don't know what the strain is going to look like, what the H and N antigens are going to be, you can't start making a vaccine against that dreaded future strain. That's what's making people crazy. And to make it worse, I'm so glad you brought that up. It turns out, you guys, our technology for making influenza vaccines, we're still using mRNA-based eggs. So the influenza vaccine strain